Hello students, welcome back to my channel. Today I am taking class for class 12th, chapter 5 CBSE board and NCERT book, molecular basis of inheritance exercises. Now here you see molecular basis of inheritance exercise. First question, group the following as nitrogenous bases and nucleoside. See, first question, nitrogenous bases, they are adenine, thymine, uracil, cytosine, and nucleosides, cytidine and guanosine, right? Next, if a double-stranded DNA has 20% cytosine, calculate the percentage of adenine in DNA. Now see, according to Chargaff's rule, in double-stranded DNA, adenine and thymine are present equal, and guanine and cytosine are in equal amount. And we know that cytosine is 20%, then guanine is also 20%. Since C and G both are 20%, so if you can calculate the total percentage of the two bases, C plus G, it will be 20 plus 20, 40%. And the total percentage of all four bases must be equal to 100. Then we can find the combined percentage. A plus T is equal to 100 minus Cg. 100 Cg is 40. Then A plus T is equal to 60 and A is equal to T. Then A plus T, this A plus T, then A plus A 60. 2A is, it is A. 2A is 60%, then A is 60 upon 2, it is the 30%. Therefore, the percentage of adenine in the DNA is 30%, right? If the sequence of one strand of DNA is written as follow 5-ATGCA23, then if the sequence of the one strand in DNA is written as the following ATGCATA, then the sequence of complementary strand in the direction adenine की जगह thymine, thymine को adenine, guanine की जगह cytosine, cytosine से guanine. Same will repeat TACG, 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 TACG with the same complementary as 3. Next one, if the sequence of the coding strand in transcription unit is written as follows, then write down the sequence of messenger RNA. अब चूंकि messenger RNA, तो इसमें क्या होगा, थाइमिन की जगह यूरेसिल आ जाएगा. If the sequence of the messenger RNA, इसमें क्या होगा 5A, थाइमिन का AUGC, 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 and third one. The beginning, it is the end. The next one, which property of DNA double helix led Watson and Crick to hypothesize semi-conservative mode of DNA replication? Explain. Now see here, Watson and Crick observed that the two strands of DNA are anti-parallel and complementary to each other with respect to their base sequences. This type of arrangement in DNA molecule led to the hypothesis that DNA replication is semi-conservative. It means that the double-stranded DNA molecule separates and then each of the separated strand acts as a template for the synthesis of a new complementary strand. As a result, each DNA molecule would have one parental and newly synthesized daughter strand, as only one parental strand is conserved in each daughter molecule. It is known as semi-conservative mode of replication. See, if this is the parental strand, then these parental strands separate and the daughter strand complementars, they develop in this. So that only parental strand conservative in each daughter molecule, that is called semi-conservative mode of replication. Now question 6 is, depending upon the chemical nature of the template DNA or RNA and the nature of the nucleic acid synthesized it, DNA, RNA, these are the types of nucleic acid polymerases. When we ask that depending on the template and synthesized nucleic acid, the nucleic acid polymerases are classified into 4 main types. First one, DNA dependent DNA polymerase, DNA replication. Second one is DNA dependent RNA polymerase, that is process transcription. RNA dependent DNA polymerase, reverse transcription. RNA dependent RNA polymerase, RNA replication. So the types of polymerases are DNA dependent DNA polymerase, that is template DNA, synthesized nucleic acid DNA, means catalyzes DNA replication, producing new DNA strands from DNA template. जैसे ये template से नया DNA strand बना लेते हैं. और DNA डिपेंडेंट RNA पॉलीमरेज, तो इसमें टेम्पलेट DNA synthesized nucleic acid RNA involved in transcription where the DNA sequence is copied into an RNA molecule. RNA dependent DNA polymerase, template RNA, synthesized nucleic acid DNA, catalyzes the synthesis of DNA from the RNA template, a process called reverse transcription. It is present in some viruses. Now the next one here I told you template, list 2 essential roles of ribosomes during translation. Acha, last one is also left, RNA dependent RNA polymerase. It is template RNA, synthesizes nucleic acid RNA, synthesizes new RNA strand is using an RNA template. This is typically found in RNA viruses and replicate their genetic material. I hope it is the previous question, class 4th part was left. RNA dependent RNA polymerase. Now here, this is the 6th one. Now the 7th one, how did Hershey and Chase differentiate between DNA and protein in their experiment while providing that DNA is the genetic material? Now see here, Hershey and Chase used radioactive isotopes to differentiate between DNA and protein in their experiment with bacteriophages and E. coli, providing DNA is the genetic material. They labeled DNA with P32 and protein with S35. By infecting bacteria with these labeled phases and then separating the phase codes from the bacteria, they found that only the radioactive phosphorus from DNA entered the bacteria, while the radioactive sulfur from protein remained outside. This demonstrated that DNA, not protein, is injected into the bacteria and carries the genetic information. Therefore, Hershey and Chase conclude that DNA is the genetic material. Here you can see over here, ये हर्सी एंड चेस, इन्होंने ये देखिए, ये P32 एंड द सल्फर 35, द रेडियो एक्टिव कैप्सूल बैक्टीरिया से जब ये इन्फेक्शन हुआ, इन्फेक्शन में आप ये देखिए प्रोटीन कोड बाहर रह गया और सिर्फ जेनेटिक मटेरियल अंदर ब्लेंडिंग जो है सिर्फ जिते, तब यहाँ पर जब इसमें सेंट्रिफ्यूलाइज हुआ तो नो रेडियोएक्टिविटी डिटेक्टेड ओवर द एयर, बट हियर डिटेक्टेड इन द सुपरनेट, नो रेडियोएक्टिविटी डिटेक्टेड इन द सल्फर से, बट हियर रेडियोएक्टिव डिटेक्टेड in the phosphorus third where the genetic material only has entered. Next question आपका question 8 है, differentiate between the following: repetitive DNA and satellite DNA, messenger RNA, transfer RNA, template strand and coding strand. Now see it here. First one, repetitive DNA and satellite DNA. Repetitive DNA is broad category of genetic material where short DNA sequences are repeated many times scattered throughout the genome. Whereas in case of satellite DNA is a specific type of repetitive DNA characterized by its highly clustered tandemly repeating sequences found at centromeres and telomere. Messenger RNA and transfer RNA. Messenger RNA acts as a template for the process of transcription. Transfer RNA acts as an adapter molecule that carries a specific amino acid to messenger RNA for the synthesis of polypeptide. It is a linear molecule. It has a clover leaf shape. Template strand and coding strand. Template strand of DNA acts as a template for the synthesis of messenger RNA during transcription. Coding strand in a sequence of DNA that has some base sequence as that of messenger RNA, except thymine is replaced by uracil, U. ये U से होगा. In DNA, it runs from 5 to 3, it runs from 3 to 5. Next question 9. List 2 essential roles of ribosomes during translation. 2 essential roles of ribosomes during translation are: first, to provide a site for protein synthesis. Ribosomes act as a platform for messenger RNA and transfer RNA to interact, facilitating and decoding of genetic information into a polypeptide chain. Second point, catalyze the formation of peptide bonds. They also contain enzymatic activity, specifically in large subunit that forms peptide bonds between amino acids, thus linking them together to create a protein. Next question, in the medium where E. coli was growing, lactose was added, which induced the lag operand. Then, why does the lag operand shut down sometime after condition of lactose in the medium? See, the lag operand shut downs after lactose in the metabolized because decrease in lactose level removes the inducer and keeps the repressor protein inactive. When the repressor can again bind to the operator, it blocks the RNA polymerase, halting transcription and the production of the enzymes needed to be further metabolize lactose. Catabolite repression influenced by glucose level can also contribute to shutting down the lag operand even when lactose is present. Here you can see with this experiment over here. Here, the lag operand. It is in the absence of the reproducer. When the reproducing this repressor RNA, then repressor bind to the operator region and prevents RNA polymerase from the transcribing the operand. But when in the presence of DNA, in the presence of the inducer, जब inducer इसमें present होगा, when inducer is the present, then what happens? Transcription occurs. This the lag, a messenger RNA synthesized from the repressor. Here it does not bind up. Here it binds up as the inducer is introduced in it. That inactive repressor, then these granule polymerase transacylatize and the translation occurs, means it changes into that one. Next one is your question number 10. In the clubbing addition to lactose ये हो चुका है. Explain in one of the two lines the functions of the following promoter, transfer RNA and exons. This one, 10th catalyzer over this, lag operand is over. Now here, promoter. Promoter is the region of DNA that helps in initiating the process of transcription. It serves as the binding site for RNA polymerase. Second, transfer RNA. Transfer RNA in small RNA that reads the genetic code present on messenger RNA. It carries specific amino acids to messenger RNA on ribosomes during translation of proteins. Exons are coding sequences of DNA in eukaryotes that transcribe for protein. Your next question, 12. Why is the Human Genome Project called a mega project? The Human Genome Project was a mega project that aimed to sequence every base in human genome. This project has yielded much new information. Many new areas and avenues have opened up as a consequences of the project. DNA fingerprinting is a technique to find out variations in individuals of a population at DNA level. It works on the principle of polymorphism in DNA sequences. It has immense applications in the field of forensic science, genetic biodiversity, and evolutionary biology. Next, what is DNA fingerprinting? Mention its application. See, DNA fingerprinting is a technique used to identify and analyze the variation in various individuals at the level of DNA. It is based on the variability and polymorphism in DNA sequences. Applications: it is used in forensic science to identify potential crime suspects. It is used to establish paternity and family relationships. It is used to identify and protect the commercial varieties of crops and livestock. It is used to find out the evolutionary history of an organism and trace out the linkages between groups and various organisms. Your 14th one, last question. Briefly describe transcription, polymorphism, translation, bioinformatics. The first one, transcription. The process of synthesizing an RNA molecule like messenger RNA from a DNA template. It is the first step in gene expression. creating a portable copy of the genetic information from DNA. For example, a DNA gene sequence is copied into a messenger RNA molecule. Second point, polymorphism. It is the occurrence. It is the occurrence. It is the occurrence of different forms, alleles of a gene or DNA segment within a population. It leads to genetic variation among individuals, which can result in differences in physical traits, behavior, or physiology. For example, the presence of multiple different versions of a particular gene among people in community. Next, translation. It is the process of synthesizing a polypeptide, a protein, from the messenger RNA sequence. It involves decoding the messenger RNA codon's 3 nucleotide sequences into a chain of amino acid, which then folds into a functional protein. For example, ribosomes read the messenger RNA and link amino acids together to form a specific protein. Bioinformatics. It is an interdisciplinary field that combines biology with computer science and information technology to manage and analyze biological data. It uses tools to extract knowledge from large biological data sets such as DNA sequences and protein structures. For example, using software to align DNA sequences to find evolutionary relationships or predict the structure of a protein. That is what all about your exercises. Any doubt anywhere, you can ask me in the comment section. Don't forget to subscribe the channel, share to your friends. Again, we will meet in the next video. Thank you.